Kaposi sarcoma, it's a cancer, it's a vascular tumor that's caused by a virus, and it affects people with and without HIV. And so the standard therapies that are available for this condition are mainly chemotherapies and some immunomodulatory agents. And so there have been very few therapies that have been explored for Kaposi sarcoma, and those that are T-cell sparing, because it's a tumor that's really driven by immune depletion, immune dysregulation. And so we were very excited to explore CDK4/6 inhibitors, which has a lot of in vitro studies using KSHV infected B cells and endothelial cells. And there has been a lot of data to suggest that CDK4/6 inhibitors might actually be, affect the virus that drives KS in these cell lines. And so we developed a study using Abemaciclib in Kaposi sarcoma in participants with both HIV and without HIV. And so that's the study that I'll be presenting at ASCO this year. So we enrolled participants with and without HIV who had confirmed Kaposi sarcoma. They were required to have at least 5 lesions on the skin, because it's a disease that affects the skin, but it can also affect internal organs. And so it's a phase 1/2 study because this drug hasn't been used in this indication before. So the first phase was ensuring that we established a viable dose. So we used the standard approved dose of 200 milligrams twice a day, orally in participants with KS, enrolled participants who'd had prior KS therapy. So mainly those chemotherapies, immunomodulatory agents that I'd mentioned. And then in the second phase, we enrolled up to 15 participants who had had prior KS therapy and 10 participants who actually had no prior systemic KS therapy. So could you use Abemaciclib up front? And so we've enrolled 34 participants in this phase overall, 31 who have been evaluable for their KS. And so our response rate so far has been 84%. So the 34 participants with KS, 25 whom have HIV. And so I found that among all participants, the response rate for KS per the ACTG AIDS clinical trial group criteria, which is a standard trial method for KS assessment, was 84%. Among those who had had prior systemic KS therapy, the response rate was 81%. And in those who had had no prior KS therapy, so in a person with HIV, they just had ART for their KS and HIV treatment, the response rate was actually 90%. So 9 out of 10 participants responded. So we're very excited about those results. And interestingly, we've also found that this therapy is actually T-cell sparing. So in a disease that's driven by T-cell depletion, again, T-cell dysregulation, this is a drug that actually doesn't affect your T-cell, CD4 T-cells. So that's been quite interesting to find as well. Yeah, so we're hoping that, given the excellent response rates, that this provides an option for participants in the future who have KS, both with and without HIV. It's often a very stigmatizing condition, especially in its association with HIV. We know that people with HIV at any CD4 T-cell count can develop KS, and so this certainly provides an additional option, again, to avoid T-cell depleting therapies. And in terms of implications, we're hoping that as we validate these findings with additional patients, participants in our advanced stage cohort at our center, we'll hope that one day this will get incorporated into clinical guidelines for Kaposi sarcoma. Well, I think it's more that, I think diseases like the ones we treat, Kaposi sarcoma, is one that was identified very early in the HIV epidemic. So this was back in the mid-late 80s, early 90s, and young men were dying of this condition. And now, it is much rarer because there are advances in HIV, but it continues to be a cancer, again, that's very stigmatizing, is somewhat left behind with many advances, and there have been few people driving these innovations. So I'm very humbled to be one member of the team that has been really trying to promote understanding of KS and KSHV pathogenesis.